One behavioral aspect of response to novelty is exploration, which involves interacting with novelty, here illustrated with novel objects for instance, to gain information about the environment. And this behavior has been suggested to be more exhibited in highly variable habitats, as it may greatly facilitate exploitation of novel resources. Since ecological variables can vary considerably from one island to another, the remote islands of the southern Indian Ocean provide useful sites for testing this hypothesis. Here, we assessed approach and touch behaviors in two subspecies of the black-faced sheathbills. But before delving into the details of how we proceeded, we first need to speak a bit more about the species itself. Although this land-based bird resembles to white pigeons at first sight, it is closely related to the Magellanic plover. Similarly to other insular species, sheathbills have few natural predators and show low neophobia. We studied two subspecies that frequent two different archipelagos in the subantarctic. The first one, Chionis minor crozitensis, inhabits Crozet Islands, while the second one, Chionis minor minor, frequents Kerguelen Islands. In the Crozet group, at Baie du Marin more specifically, sheathbills breed near a colony of king penguins in a highly variable environment where food is scarce during winter. They feed on various resources such as eggs, small chicks, fish, carrion, and other items like feces and cell placenta. In this group, sheathbills also very often practice kleptoparasitism by opportunistically stealing food from penguins, regurgitating food for their chicks. In the Kerguelen group, at Ilvert more specifically, the environment is less variable, which is due to the broad intertidal zone allowing sheathbills to feed all year long. Over there, they are also very opportunistic and forage on various items such as intertidal alga, carrion, insects or mussels. At this location, breeding individuals also use plants, rib mussels, blue mussels and limpets as decorating objects that they carry and place in and around their nest cavity. So comparative investigation with this model system may thus be relevant to help determining the ecological variables that predict exploration behavior. We thus field tested whether sheathbill breeding pairs approached novel objects resulting from the interaction between attraction and avoidance, and touched novel objects reflecting a propensity for exploration. First, we expected that low neophobia resulting from reduced predation pressure related to insular environments should favor approach behaviors in both groups. We also expected that high environmental variability should elicit faster approaches, but also more explorations in sheathbills from the Crozet group. 12 wild established breeding pairs of black-faced sheathbills participated in this experiment during November-December 2021. 6 pairs in the Crozet group and 6 pairs in the Kerguelen group. We used a simple and well-established method for investigating exploration in sheathbills, which consisted of presenting one novel object on the ground within each pair's territory. Since breeding pairs are very territorial, this allowed us to test pairs separately in the field. We used 3 novel objects that varied in color, shape and size: an orange wooden plank, a Ziploc plastic bag filled with black sand to prevent birds from flying off with it, and a yellow buoy. When both individuals of the focal breeding pair were present on their territory, the experimenter triggered the camera, slowly approached the breeding pair, placed the novel objects in front of and at about 1 meter distance from the birds, and retreated at least 10 meters. Only the breeding partner that first approached the novel objects was included in the analysis, and the order of presentation of novel objects varied randomly across breeding pairs. Each trial started when the experimenter left the test area and ended after the maximum trial duration had elapsed. To assess prediction 1, that is whether the two groups significantly differed in their propensity to approach the novel objects, we built a generalized linear mixed effects model with a binomial distribution. The variable response was coded as 0 when no approach occurred and as 1 when approach occurred. Regarding prediction 2, whether approach latencies significantly differed between the groups, we also used a GLMM but with a gamma distribution, since our response variable was latency in seconds. Finally, to test whether the two groups significantly differed in their propensity to touch novel objects, that is prediction 3, we used a GLMM with a binomial distribution. The response variable was coded 0 when no touch occurred and as 1 when touch occurred. All breeding pairs on both islands approached the novel objects, demonstrating low levels of neophobia. Contrary to our second prediction, we found no differences between the two subspecies in the latency to approach novel objects. In accordance with prediction 3, however, the Crozet group touched significantly more the novel objects compared to the Kerguelen group. Finally, contrary to other avian species, stimulus typology, that is the difference between objects' property, had no influence on sheathbill's approach or touch behaviors, at least among the objects we used. Overall, high environmental variability is a suggested explanation for the source of variation in response to novelty between the Crozet and the Kerguelen groups. However, caution in interpretation is required. First, because our study focused on a limited number of populations, and also because these group-specific differences in exploration may also have resulted from other or a combination of influencing factors. For instance, since these two subspecies and all four subspecies in Canis minor generally are sedentary and possess poor flying abilities, they do not disperse at other islands, although there can be a short distance to cross between sites. One may thus argue that the difference between our two subspecies is not due to environmental but rather genetic variability. But this seems insufficient to explain our results. There is indeed a level of heterozygosity 3 times higher at Kerguelen than Crozet, which may be due to environmental factors such as habitat diversity. Comparing Kerguelen and Crozet subspecies was justified by the fact that the broad intertidal zone is only present at Kerguelen Islands. Future studies may compare Kerguelen populations in penguin areas which still have the possibility to feed on the intertidal zone to see if an intermediate value between low and high exploration is obtained. Importantly, interaction with humans and or exposure to anthropogenic stimuli may promote exploration in some species. In one of our studies conducted on wild subantarctic squaws, we raised the possibility that wild squaws, which had interacted with a human experimenter during food-rewarded behavioral and cognitive experiments, may have developed an increased tendency to explore novel objects presented by humans. Squaws and sheathbills cohabit with humans and face histories of exposure to them in many locations. This may give rise to similar levels of motivation to gain information through interacting with novelty. Since 1964, sheathbills at Crozet have had many opportunities to interact with humans who visit daily the permanent research station inside the island's king penguin colony. Over there, sheathbills often try to steal objects or food from scientists' belongings. Although further investigations are needed, such experience may have reinforced sheathbills' attraction to humans and exploration tendencies through associative learning of the relationship between food and humans. Crucially, knowledge about the behavior of endangered species is fundamental to developing relevant applications for conservation purposes. Although sheathbills are not classified as endangered species per se, global warming combined with the introduction of alien species have caused declines at many locations. For instance, feral cats have been quite detrimental on breeding populations, and house mice have indirectly impacted sheathbills by depleting terrestrial invertebrates. At Île de la Possession in Crozet Islands, rats are the only introduced predators, and they have had a negative impact on seabirds. In some parts in the Indian Ocean subantarctic, eradication plans have been devised to wipe out introduced rats using novel objects such as brightly colored food poisoned pellets. Since we now know that sheathbills tend to touch unfamiliar colored items at Crozet Islands, future conservation measures of this kind will need to be managed with caution.